Welcome, my dear students. Today we are going to explore one of the most important concepts of classical genetics, that is linkage and crossing over. You have already known that genes are assorted independently, but in reality, genes located on the same chromosome often travel together, showing linkage phenomena. So during meiosis, crossing over breaks this linkage and creates new gene combinations. In this class, we will clearly understand what is complete and what is incomplete linkage. What do you mean by 3-point cross and linkage mapping construction, linkage map construction, coefficient of coincidence and interference, and the molecular mechanism of recombination explained by Holliday Model in 1964. These concepts that are extremely important for your undergraduate and postgraduate university exams, CSIR-NET exam and Life Science GATE exam. So let's uncover how genes are mapped and reshuffled inside the chromosomes. So concept of linkage. Linkage, basically we know that each chromosome contains many genes arranged linearly on the chromosome. So this is the arrangement of genes on the chromosome. So genes may be located on different chromosomes. They are short independently. We have studied from the Mendel's law. We know that. And on the if the genes are present on the same chromosomes, they tend to be inherited together. So linkage is the phenomena where two or more genes located on the same chromosome show non-random assortment during meiosis. So definition, so linkage is the tendency of genes present on the same chromosome to be inherited together due to their physical association. Now, we should learn about the characteristic features of linkage. It involves two or more genes located on the same chromosome. So, genes are arranged in a linear order. They can involve dominant genes, recessive genes, and combination of dominant and recessive genes. So, they are closely located genes show stronger linkage. Linked genes always produce more parental types and fewer recombinant type. The test cross ratio here deviates from 1 is to 1 is to 1 is to 1. So the linkage may involve all desirable traits, all undesirable traits, and mix of both. So important cause of genetic correlations beside pleiotropy. So strength of linkage is inversely proportional to the distance between genes. Maximum number of linkage group is the haploid chromosome number of that particular individual. So linkage can be broken by crossing over and repeated intermating over generations. So chromosome theory of linkage, which was proposed by Thomas Morgan. So Morgan and Cassell, they jointly proposed that linked genes are located on the same chromosome. Genes are arranged linearly and linked genes remain physically associated during inheritance process. So distance between genes determine the strength of linkage. Short distance indicates strong linkage and long distance indicates weak linkage. So types of linkage based on the crossing over event. Complete linkage, here no crossing over occurs and genes are inherited together generation after generation. Only parental combinations appear. Example, Drosophila melanogaster genes for bent wings or BT, 7 bristles or SVN, which are located on chromosome 4th. Key features. Complete linkages features include there should be no recombination and the test cross shows only two phenotypes. Incomplete linkage, the crossing over occur occasionally. It produces both parental and recombinant type. Recombinants appear in small percentage. So examples include maize or Gia maize genes like colored seeds, full endosperm, colorless and shrunken. So results, parental combination is 96% in case of incomplete linkage and recombinants are 4%, which indicate complete linkage with crossing over. Based on genes involved, coupling or cis linkage, dominant genes together or recessive genes together, example capital A capital B or small a small b. Repulsion or trans linkage, the dominant genes linked with recessive gene, examples capital A small b or small a capital B. Based on chromosome involved, autosomal linkage, when the linked genes are present on the autosomes, they are called as autosomal linkage, and when the linked genes are present on the X chromosomes, they are called as X chromosomal linkage. So what is linkage group? All genes on one chromosome form one linkage group. So the number of linkage group is equivalent to the haploid chromosome number. Crossing over. What is crossing over? Crossing over is basically the exchange of genetic material between non-sister chromatids of homologous chromosomes during prophase 1 of meiosis, resulting in new combinations. So this is the occurrence of crossing over, and it produces the recombinant chromatids, resulting in new gene combinations. So stages of crossing over, it occurs during Pachytian stage of prophase 1. It requires synapsis, synaptonymal complex, etc., and it is visible as chiasmata. This is the chi or Greek X-like structure which is known as chiasmata. So molecular basis of crossing over, Holliday model 1964. The structure is named after Robin Holliday, the molecular biologist who firstly proposed about the molecular steps of molecular basis of crossing over in 1964. According to his concepts, the steps are following. Double-stranded break occurs in DNA. Exonucleases start remove nucleotides from 5' ends. Formation of single-stranded overhang. Strand inversions occur into homologous DNA. DNA synthesis using homologous strand as template. Formation of Holliday junction, that is a cross-shaped structure is formed, and resolution of junctions by enzyme like MSH4, MSH5, etc., which results in recombinant chromatids, and gene conservation sometimes can also occur. Significance of crossing over, we know crossing over produces genetic variability, prevents accumulation of harmful alleles, maintains useful gene combinations, and they are essential for microevolution and linkage map construction. They help in plant and animal breeding. Relationship between linkage and crossing over. Linked genes may still recombine due to crossing over, so their frequency of recombination is in proportional to the distance between genes. Crossing over weakens linkage and produces recombinants. These are the comparison between crossing over and linkage. Construction of the linkage map or 3-point test cross. Principle uses recombinant frequencies to determine gene order and distance between genes. Steps: cross, triple heterozygote versus triple recessive. Identify parental types which show higher frequency, and double crossover which show lower frequency. So determine gene order or double crossover types or DCOs. Calculate map distance. Map distance is cm, number of recombinants by total number of offspring into 100. So one map unit is equal to 1 centimorgan, is equivalent to 1% recombination. So coefficient of coincidence C, C is the observed double crossover value, and it is expected double crossover. This is the ratio of observed double crossover and expected double crossover. So C, what is interference? Interference I is equal to 1 minus C, that is it measures how much crossover reduces the probability of another nearby crossover. Type, it can be positive interference when fewer double crossovers, and negative interference causes more double crossovers. So linkage and pleiotropy. In number of genes, in case of linkage there should be more than or equal to 2, and in case of pleiotropy it will be 1. Cause, physical proximity is the reason for linkage, and single gene effect is the example the cause of pleiotropy. Can be broken? Yes, linkage can be broken by means of crossing over, but pleiotropy cannot be broken. Identification, recombinants possible, and in case of pleiotropy there is no recombinants. So significance of linkage in plant breeding. Effect on selection, desirable genes are linked, advantageous, and desirable plus undesirable linked genes causes difficult selection. Effect on genetic variation and repulsion linkage. Dominance variation will be increased, and additive variance will be decreased. So effect on genetic correlation, linked traits show high co-heritability, helps in simultaneous improvement of traits. So as we conclude, remember that linkage and crossing over together explain how genes are inherited and reshuffled on chromosomes. From understanding gene order through linkage mapping to measuring recombination using interference and coefficient of coincidence, and finally the molecular exchange of DNA strands by the Holliday model 1964. Each concept reveals the dynamic nature of heredity. These ideas are essential for mastering classical and molecular genetics for UG and UG, PG, and CSIR-NET or competitive exams. So keep revising and exploring how chromosomes shape variation and evolution. Thank you.